Good morning, students. Today in this session we are going to discuss about NEET and K-CET level coaching part. On the chapter Cell the Unit of Life. Subtopics: Prokaryotic cells, cell envelope and its modifications, ribosomes and inclusion bodies. Right? Now let's enter into today's session. First question. Choose the irrelevant regarding prokaryotic cell from the given. Option A, PPLO, B, blue-green algae, C, mycoplasma, D, human sperm cell. The correct answer for this question is option D, human sperm cell. Now let's discuss about this question in detail. PPLO stands for pleuropneumonia-like organisms. Blue-green algae, it is also popularized as cyanobacteria. Mycoplasma, it is also considered as the smallest living cell, right? First three cells, PPLO, blue-green algae, mycoplasma, are not having well-defined membrane-bound organelles in the cytoplasm. So they belongs to prokaryotic cells category. Next, human sperm cell is having well-defined membrane-bound nucleus in the cytoplasm. So this human sperm cell is related to eukaryotic cells category. So the irrelevant option for this question is human sperm cell, option D. Have you understood? Now moving to second question. Identify the correct option about prokaryotic cell. Option A, they are smaller. Option B, multiply more rapidly than the eukaryotic cells. Option C, vary greatly in shape and size. Option D, all the above. So students, prokaryotic cells are generally very small and microscopic in nature. They are smaller than even eukaryotic cells. Remember this point. Then they can rapidly multiply than the eukaryotic cells. And they also greatly vary in shapes and sizes. So all the ABC options are related to prokaryotic cells only. So the correct option for this question is option D. Right? Now, hope you understood this question. Moving to third question, matching type. So this is very important way of asking for competitive examinations like NEET and K-CET. The same method, the same question they have asked in previous NEET examination also. Now let's discuss. Column 1 should match with column 2. Understand? In column 1 they have given different shapes of bacteria. In column 2 they have given different types of bacteria. Now let us see column 1. Rod shape. Which is rod shaped bacteria in column 2? It is generally bacillus. So A is matching with the Roman number 4. Right? Next. B, spiral shape. Spiral shape is generally in column 2, spirillum bacterium is having. So this is also popularly called as ribbon shaped bacterium. So that's what B is matching with the Roman number 1. Next, comma shape. Comma shape is generally exhibiting in column 2 by Vibrio bacteria. So this C is matching with the Roman number 2, right? Next, last one, spherical shape. Spherical shape is also called as round shape. The round shaped bacteria in column 2 is nothing but, which is that the last one, coccus. So Roman number 3 is matching with the D. So the correct option for this question is option B. I repeat once again. A is matching with 4, B is matching with 1, C is matching with 2, D is matching with 3. Hope you understood this question explanation, very important. Now moving to fourth question. Choose the correct option for polyribosomes formation. So polyribosomes formation is very important event in the ribosomes. Now let's talk about options. A, ribosomes attaches to single mRNA. Option B, ribosomes attaches to single rRNA. Option C, ribosomes attaches to double stranded DNA, shortly called as dsDNA. Option D, ribosomes attaches to single stranded DNA, shortly called as ssDNA. So here in this one point I would like to bring to your notice formation of polyribosomes. Let's see explanation. Poly means many ribosomes. Many ribosomes in the cytoplasm attaches to the single mRNA, full form messenger RNA, and forming polyribosomes are also called as polysome. So the correct answer for this question is option A. Have you understood? Now moving to fifth question. Identify the genetic material in bacteria. Option A, genomic DNA. Option B, circular DNA. Option C, both A and B. Option D, none of the above. Generally in bacteria the genetic material is quite unique since it is prokaryotic cell compared with the eukaryotic cells. In eukaryotic cells the genetic material is generally DNA. It is well elaborate, but in prokaryotic cells it is quite unique. Let's see options. A, genomic DNA. B, circular DNA. C, both A and B. D, none of the above. Genetic material in bacteria, genomic DNA, and as usual circular DNA also. So with the A and B options, C is the correct option for this question. Understand? Now coming to sixth question. Smaller DNA is called as option A, polysome. Option B, plasmid. Option C, transmit. Option D, all the above. So the correct answer for this question is option B, plasmid. It is the smaller DNA. So fifth question and sixth question regarding genetic material of bacteria, we have learned about genetic material in regular theory class. Hope you remember this one. Right? Now moving to seventh question. Name the special membranous structure which is formed by the extinctions of plasma membrane into the cell. Option A, polysome. Option B, ribosome. And option C, mesosome. And option D, all the above. So here I would like to bring to your notice very important information. In bacteria, special membranous structures are presented. Those are the extensions of plasma membrane into the cell or towards the cell. Understand? Those are called as mesosomes. Very important for PU board examinations also. Understand? So the correct option is option C. Next, eighth question, statement based. This is very important way of asking for competitive examinations, especially from cell chapter. So now let us talk about the question. Choose the correct statement from the given statement 1 and 2. Statement 1. The cell envelope in bacteria consists of a tightly bound 3-layered structure. Statement 2. The outermost glycocalyx followed by the cell wall and then the plasma membrane. Students, I would like to tell you our important point. In regular theory class, we had a discussion regarding the structure of bacteria. Bacteria consists of tightly 3-layered structure. It is called as cell envelope. It is tightly bounded with each other. Even though tightly bounded with each other, each layer has its own specific function. Keep this point in the mind. So those 3 layers as follows. Outer one, glycocalyx, and middle one is called cell wall. The innermost one is plasma membrane. So according to the given statements, both the statements are correct. Understand? So which option is correct for the given question? Option C. Both the statements are correct. It means the cell envelope in bacteria consists of a tightly bound 3-layered structure. Statement 1. And statement 2 is the outermost glycocalyx followed by the cell wall and then the plasma membrane, which is also called as cell membrane. So both the statements are correct. So option C is correct for this question. Next, ninth question. The loose sheath-like structure in glycocalyx is called option A, capsule, option B, slime layer, option C, thick layer, and option D, mesosome. So loose sheath-like structure in glycocalyx, which is the outermost layer in the bacteria, is called as slime layer. Then one more layer is present, capsule. Capsule is very tight and toughly present. So that is the specific characteristic. It is very thick and tough layer. It is nothing but capsule. Understand? So loose and loose sheath is called as slime layer, option B. Understand? So the correct answer for this question is option B. Hope you understood this explanation. Now moving to 10th question. Find out which is not the function of the cell wall from the given. So we need to identify which is not the function of cell wall. Understand? Cell wall performs some specific functions. Let us see options. It determines the shape of the cell. It is correct. B. It provides a strong structural support. This is also correct. C. Prevent the cell from bursting or collapsing. This is also correct. It's a living rigid structure. Actually, cell wall is the outer non-living rigid structure. It is not a living rigid structure. Have you understood? So, option D is not the function of cell wall. It is irrelevant to the functions of the cell wall. So the correct answer for this question is option D. Understood? Now 11th question. Pick out the semi-permeable membrane from the given. Option A, cell wall, B, plasma membrane, C, glycocalyx, D, slime layer. Students, here let's see explanation. Semi-permeable nature. What is meant by semi-permeable nature? Semi-permeable nature is also called as selectively permeable. Plasma membrane performs a specific function. It selects the liquids or fluids or even minerals, whatever required to it only. So that nature is also called as selectively permeable. It is also called as semi-permeable nature, performing by the plasma membrane. Understand, this is most important for competitive examinations. Remember it. So the correct option for this question is option B. It is plasma membrane performing semi-permeable nature. Next, 12th question. Bacterial flagellum is not composed of this following part. You know, flagellum is the locomotory organ in bacterium. This flagellum has specific parts. Understand? Now let us see options. A, filament, B, hook, C, basal part, D, middle part. So one is not in the part of flagellum of bacteria. Which is that? It is nothing but middle part. Middle part is present in the sperm cell. Understand? Remember, filament, hook, basal part, these three are the parts of the bacterial flagellum which helps in locomotion. Understand? So option D is irrelevant for the parts of the bacterial flagellum. But where we can see this middle part, the middle part can be seen in the parts of the sperm cell, human sperm cell. Right? Now moving to 13th question. Identify the different mesosomes forms from the given below. As we learn in above question, a special membranous structures which are extending from the plasma membrane into the cell or towards the cell center, they are called as mesosomes. Generally, these mesosomes are vary from one to another. They will be in different forms. Let's see what are those. A. Vesicles. B. Tubules. C. Lamellae. D. All the above. As we discussed just now, the mesosomes are available in different forms. Those are as follows: vesicles, tubules, lamellae. So with those ABC, option D, all the above is correct. Understood? Now, moving to 14th question, choose the functions of the mesosomes. We need to identify the functions of mesosomes. This is very important area for asking competitive questions because mesosomes performs different kinds of functions. First of all, let us see options, then we will discuss in detail. A. Cell wall formation. B. DNA replication. C. Respiration. Option D. Both A and C. So, choose the functions of the mesosomes. Which are the functions of mesosomes? First one, cell wall formation. It is the function of mesosome. Then respiration is also function of the mesosome. So with A and C options, option D, both A and C are the correct options. Nothing but option D. Then B is there, no? RNA replication. RNA replication is not the function of mesosomes. The correct one is DNA replication. Apart from these functions of mesosomes, there are many functions are there. Let's talk some more functions of the mesosomes for your extra knowledge. DNA replication and distribution to daughter cells. This is most important function of mesosome, and they also helps in secretion processes and to increase the surface area of plasma membrane and enzymatic content. Understand? So these are the different functions of mesosomes. In theory class, might be you have remembered this concept regarding the functions of mesosomes. Understand? So the correct option for this question is option D with A and C options. Cell wall formation and respiration. Understood? Now 15th question, matching type. This is very important way of asking. Now let us match column 1 with column 2. In column 1 there are different phenomenons are present. Let's match with column 2. Secretion processes. Secretion process is the function of, just now we had a discussion, mesosomes. It is producing some enzymes, enzymatic content to increase the surface of the plasma membrane in bacteria. Understand? So A is matching with the Roman number 4. Roman number 4. Next, cyanobacteria. Cyanobacteria is consisting a special structure. Those are nothing but helps in different events. What are those? Nothing but chromatophores. They helps in photosynthesis process by consisting colored pigments. Chromatophores are nothing but colored pigments present in cyanobacteria, helps in photosynthesis process. As we learn, another name for cyanobacteria is nothing but blue-green algae. Blue-green algae performs photosynthesis, right? This part already we had a discussion in lower chapters. So B is matching with the Roman number 3. Next, staining procedure. Staining procedure of bacteria was discovered by Christian Gram. The scientist name is Hans Christian Gram. The full name Hans Christian Gram. So C option is C is matching with the Roman number 5. The full name Hans Christian Gram. What is this staining process? Already we had a discussion in regular theory class, right? The bacteria can be separated by a staining process called as Gram staining process into Gram positive bacteria into Gram negative bacteria, discovered by Hans Christian Gram. Understood? Next, bacterium locomotion. In bacteria, the locomotory organ is nothing but flagella. Just now we had a discussion. Flagellum singular has mainly 3 parts. What are those? Basal part, hook and filament. These are the 3 parts which presents in flagellum. Next, inclusion bodies. Inclusion bodies are nothing but rigid materials in the cytoplasm of bacteria. They are present in the cytoplasm, they are freely present without depending on other, and that too they are non-membrane bound structures. So inclusion bodies, E is matching with the Roman number 1. So the correct option for this question is option D. So once again, let's discuss secretion processes by mesosome. Next, cyanobacteria has chromatopods. Bacterium locomotion by flagellum. Next, staining process discovered by Hans Christian Graham. Inclusion bodies are nothing but rigid materials in the cytoplasm of the bacteria. So A 4, B 3, C 5, D 2, E 1. So the correct option is option D. Understood? This is very important for NEET exam. Next, 16th question. Identify the structure in the bacterium which do not play a role in motility. So students, just now we had a discussion parts of flagellum, and there are few structures here which do not play a role in locomotion of bacterium, but they are present on the surface of the bacterium. Let's see what are those. Option A, pili. Option B, fimbriae. Option C, cilia. Option D, flagella. So the correct option for this question is option B, fimbriae. Fimbriae are brush bristles like structures which presents on the surface of the bacteria along with the flagella. But flagella helps in locomotion of bacteria, but fimbriae do not help at any cost for locomotion of bacterium. But what is the role? What is the use of fimbriae? They are just like brush bristles like structures. They help to hold the substratum or support. Understand? For example, E. coli bacteria, it has fimbriae. It attaches to the intestinal walls in the small intestine. Next, this fimbriae helps to even attach to the rocks in the underground water. Next, 17th question. Pick out the irrelevant one regarding prokaryotic cells, ribosomes and their subunits. Option A 50S, option B 30S, option C 70S, option D 80S. So we need to identify irrelevant regarding prokaryotic cells, ribosomes and their subunits. You know students, I will explain this part very clearly. Now listen to me. In prokaryotic cells like bacteria, the subunits are 50S and 30S. So A option, B option are correct, and together forms as a single unit it is nothing but 70S. So 70S type of ribosomes are present in the prokaryotic cells, example bacteria. So 80S is the irrelevant. 80S type of ribosomes are present in eukaryotic cells like human sperm cell and human egg cell, etc. So the irrelevant option for this given question is option D, 80S ribosomes. S stands for sedimentation coefficient or Swedberg units. Understand? Now, 18th question. Choose the organelle which is the site for protein synthesis in a cell. This is very important area for asking competitive questions. In many previous years they asked the same question. Option A, polysome. Option B, ribosome. Option C, Golgi complex. Option D, mitochondria. So there is a one cell organelle in the cell which is also popularly called as protein factory. Understand? So the cell organelle is the site for protein synthesis. Let's see options. In the options, the correct option for synthesizing proteins, it is nothing but ribosomes. Understand? As we discussed, ribosomes are present not only in the cytoplasm ribosomes are also present in few other cell organelles like mitochondria, chloroplasts in plant cells, and endoplasmic reticulum. Especially in the endoplasmic reticulum, on rough endoplasmic reticulum also ribosomes are present apart from cytoplasm. This is also a very important event for asking competitive questions. Please remember it. Next, 19th question. Pick out the inclusion bodies from the given options. Option A, phosphate granules. B, cyanophysin granules. C, glycogen granules. Option D, all the above. So the correct answer for this question is option D. ABC options, it means phosphate granules, cyanophysin granules, glycogen granules, all these are called as inclusion bodies only. As we discussed just now, inclusion bodies are reservoir materials in prokaryotic cells, are stored in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and lie very freely in the cytoplasm. Understand? So the correct option for this question is option D with ABC options. Hope you understood explanation for this question. Next, last question in this session. Blue, green and purple and green photosynthetic bacteria consists of option A, chromatophores, B, gas vacuoles, C, food vacuoles, D, excretory vacuoles. So the correct option for this question is option B, gas vacuoles. As we learned in previous class, I just wanted to give you extra information about this one. Gas vacuoles are present in certain members of algae. Those are blue-green and purple and green photosynthetic bacteria. Their role is in storing the gas and exchanging the gas from atmosphere to into the cell, from inside cell to the atmosphere. Nothing but respiration, right? So hope you understood. The correct option for this question is option B. So students, with this 20th question, I conclude today's session. Hope you understood this today's session. Thank you one and all.